Dr. Catanzaro, thank you so much for taking the time to visit with me again. I have greatly enjoyed, the relationship and following along with what you're doing. And so, thank you for being here as we dive into a really important topic.
And thanks, Logan. Thanks for having me on again. It's really it's a pleasure just being here with you and jawing the problem.
Because we have a problem, my friend. We have a problem. So let's start as we get into this conversation. Because we're going to get in the weeds in some areas, but for the basics, we've heard a lot of whether it's on social media, it's different types of conversations around problems around COVID or mRNA vaccines and all of these things is the spike protein, the spike load, spike, right? So, do you care to go into that basic, fundamental layman's terms on what is a spike protein and why is it a problem?
Yeah, the synthetic SARS-CoV-2, which is a recombinant virus, right? Which means it was a constructed virus. It's not a naturally occurring virus. It was a virus that was taken and recombinant engineering, and it was basically to discover what the resilience and virulence of this virus would be. And so the spike protein is actually part of the viral matrix. So it's a natural, it's a protein that that's pretty much expressed by the synthetic recombination that is known to create more accelerated signals to cause a more fast and furious replication of the virus itself. And so the viral ecology steps up its rate of expression in the human body, right? So there's a difference between the spike protein, viral spike protein, and then the spike associated from the vaccine, the mRNA vaccine construct, because the spike from the mRNA vaccine construct is a nucleotide segment of a spike that is uniquely different in its presentation than what we see on the recombinant synthetic spike of the virus, right? And and but the whole purpose of the mRNA vaccine related spike is to allow for a much more enhanced expression of spike to elicit an immune response, which which basically it backfired. So instead of eliciting an immune response, it winds up eliciting all kinds of what we call transcription irregularities. So we wind up actually seeing some significant amount of gene signal pathways being affected, where we've actually detected thousands of proteins affected by this aberrant transcription, right? Because the spike protein is kind of it's a rogue element. It's an element that hijacks the internal machinery of the cell, namely the ribosomes. So the ribosomal fidelity is then compromised. So anything then goes. If your ribosomal fidelity is compromised, then you wind up developing fragment proteins that are destructive and have nothing to do with body function. They have everything to do with creating more destructive action of the cells and the systems that they regulate, right?
Yeah. So for just a super basic, this is part of a viral spike protein to make more of the virus, right? This would be a
Yes.
part of a virus that goes into the cell basically to then make more viruses. But in the at least theory with the mRNA vaccines, they put in a mRNA, which is messenger RNA, to create this spike protein, so the immune system would attack it, and in theory, you would be able to be protected from whatever virus it is. In this case, SARS-CoV-2, right?
Yeah. Yeah.
Okay.
That's correct.
So what
Yes.
what seems to be happening in a lot of the long COVID, cancers, on and on and on, right, is that we're seeing elevated levels of spike protein. And so what is that meaning for a clinician?
So what that means when you see elevated spike protein, that means the retention of spike within the human tissues is it continues way past infection and actual vaccination. So you wind up actually having more retention, which uh, creates more risk for transcription errors to occur, like we've been seeing because of that retention. And what winds up happening is you get concomitant inflammatory responses that are hyperinflammatory responses due to immune dysregulation or immune incompetence, if you will. So the immune incompetence part of it is a very real thing, because what we wind up seeing is elevated antibodies going up for specific to spike, and those antibodies continue to rise, and they continue to rise. And a lot of the times what you wind up seeing is, and a lot of people might be aware of the fact that if you are infected with a virus, you might have antibody levels that stay elevated for a long time, right? And they might continue to elevate. So the risk for cross reactivity of these antibodies is higher. So once you actually have this posture of hyperinflammatory immune dysregulation, then the risk for autoimmunity goes up and destructive autoimmunity goes up. And then, of course, systems are affected in a very negative way, right? So, so instead of getting immunity, you get immune incompetence.
It's just chaos, right? And so I think a couple other ways that I have at least been looking at it is like inflammation and chaos are essentially the same thing, right? There's this lack of coherence, a lack of fidelity in all of the signals that are going on. So to me, through this little bit of a biophysics lens, what what seems to be the case is that the spike protein is an electromagnetic nightmare in of itself too, right? And so all things are thrown off in this whole other context that I don't really hear anybody else talking about it in more of like the mainstream alternative, for lack of a better term, spaces is not on this biophysical aspect. But before we hit record, we were talking a little bit about the mitochondrial function. So that would be, this is talking about the electron transport chain, right? NAD to oxygen and ATPase, right? So like what are we seeing in the mitochondrial dysfunction with this spike protein, which I would go out on a limb and say is the majority of our chronic diseases are related to the mitochondria.
What we're actually seeing is an extreme amount of reactive oxygen species activity in the mitochondrial matrix. So what happens is that the amount of load of mitochondrial stress it raises up, in an exponential way. So that means that the stress-related activity on, the ETC ability to generate activity and energy, the ATP ability goes down. A very significant amount of depletion happens. The mitochondrial complex becomes very loose and less tightly wound as a protein, and so there's misfolding that occurs on that protein, right? So when you talk about biophysical influences, you're talking about the actual effects on the folding capabilities of the protein to maintain its intact structure. So what you wind up getting is a loosened effect that creates a very big disturbance. And you know that complex 5 is a real big part of mitochondrial function. But here's when the mitochondria start to minimize their ability to generate the activity on a regular. So in other words, you got the homeostasis of the mitochondrial controls. Then what happens is then there's like there's like a limp along type of situation that happens with the crosstalk between the mitochondria and the ER, the endoplasmic reticulum, right? So any of these internal related stressors that you see that affect this ribosomal interface, that then creates more of a related, anything goes switch. Like in other words, we no longer have a grip on what we're supposed to be translating here. Now our message is garbled pretty severely. So now the mitochondria fall apart, the ER falls apart, the ribosomes don't know how to transcribe and translate the right message. There's a whole lot of interface, and that itself then creates a heavy risk on DNA itself. So then that's when you start finding that the DNA related stress on checkpoint controls go out the window. So that means that DNA then has the risk for double-strand breaks, and then that's when you wind up seeing the potential for integrative events to occur in the human host. So like in other words, the nucleus then becomes involved And that's when you start getting replication of this bad message in other cell lines, right? So that's a very simple way to explain it, because we have been seeing genomic integration evidences to where we picked up a recent translocation, a host vector translocation that fused into chromosome 19. Chromosome 6 and chromosome 19 are two very important. All your chromosomes are important. So a fusion that occurs anywhere is a problem. Chromosome 6 and chromosome 19 share some elements of immune controls and suppressing oncogenic signals. So all of these different things that are happening out there where people are talking about turbo cancers, the reality is that because of what we see in this spike initiated encoding dysfunction or irregularities, the risk goes higher and higher with each new cell line that is developing to produce a corrupt or garbled message. And with that, then the risk of integrated events occur as well, because we're talking about spike retention, spike initiated transcription translation errors, and we're talking about replication issues in the billions of copies. So something that our bodies are very intense engineering. We're not talking about one by one by one by one. We're talking about billions of duplications, right? And that's what we're dealing with at this point.
So, going back to mitochondria, essentially what I took away from that first part that you laid out is that we are raising the heteroplasmy rate of the mitochondria, based off what Dr. Doug Wallace is put out, and so that is going to also lead to transcription errors within mitochondrial DNA. So that sounds like
Yes.
a complete catastrophe in of itself. And then so there we're talking about different chromosomes and 19. What I have found pretty early on was the SV40 aspect, and this is going back to the full SV40, right, with polio vaccine and the monkey kidneys and all that. So what it seems like to me is it disrupts the T antigen, right? Like it suppresses our P53. So when you're talking about these issues, is that kind of a similar mechanism that you're seeing for tumor suppression?
Yes, I mean it is involved, right? Because when we talk about the SV40, in the research environment, there's certain plasmids that are used as a means of regular research, science, and development. And so you've got DNA plasmids and there's higher plasmid motifs that researchers go to, right? And when we talk about that they're going to naturally go to a plasmid that's going to actually have a greater effect on expression than they would to use a plasmid that's less of. There's a whole library of plasmid DNA plasmids, believe me. It's a sea out there. If you looked into it, you would see that there's quite an exhaustive catalog. But the thing about it is, when we're talking about SB40 and we're talking about the likelihood of creating what we call DNA compromise, this is exactly what we see. We're seeing a compromise on the integrity of the ability for a regular what we call uh, natural DNA engineering to remain its intact, stable double helix structure, right? And and these contaminants like SB40 are definitely involved in that. So we wind up seeing a relationship between the actual nucleotide element interface along with the integration events. So, like for instance, this integration event that we detected is on the is outside of the AAVS1 location, which is known as a safe harbor on chromosome 19, and it's outside of that. So, in other words, it is in lethal territory. It's in a very pathogenic, high risk area. So, more likely to have errors to occur from a standpoint of whatever that band controls and signaling, right? And And in this case, in a lot of these turbo cancer cases, and we're going to be seeing more and more of this relationship. And we may not just see it on chromosome 19. We might see it on other chromosomes. Might see it on chromosome 2 or 6 or 8. It's hard to say. These fusion translocations, because of the way the vaccine companies designed this, they mimicked this as close to the human genome as possible, so that you would not be able to detect that it's other than human genome. So, the thing is that the detective work the surveillance work has to be greater. The burden of greater surveillance is necessary. So before any of these technologies can take hold, including CRISPR and circular RNA and endless RNA and self-amplifying RNA, all of these technologies that are developing have no, they actually have no surveillance safeguards. You have to have molecular surveillance safeguards. You just can't put something in a system and just cross your fingers and think that it's going to do what you think it's going to do, and or whatever your intention is in the engineering that is potentially harmful, right? Nobody wants to think that anybody has has the want to create harm. But for all intents and purposes, if you look at this, there is a great degree of negligence here when it comes to the human genome, because we don't actually have good safeguards. There's no safeguards. There's no surveillance, and we need to have these this has to be part of the system's infrastructure to assure that these technologies are moving in a very positive direction and not in a harmful one.
So correct me if I am wrong on absolutely anything, but it is my understanding that it was like over 6 billion people got some form of an mRNA vaccine. At least once. There is a compounding effect. The more you got, the worse that it tends to be, and that we are seeing infertility rates just being out of control. The cancers. Everybody's heard of myocarditis, but that's like not even it, right? Like a kidney dysfunction, there's kidney failure, like on and on and on, right? And so and
Yes.
and 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 kids are dying. And we're still popping them. And so like the whole aspect of this just kind of blows my mind. So I guess, John, what I'd really like to know is this integrated for good? Because that's important, right? Like I don't want to skirt around that. Does this integrate into the DNA?
Does it integrate into
The DNA
the
just permanently.
Well, what we're seeing is evidence is pointing in that direction, where there's integrated events happening in the host genome. And that's an alarming find. That's very alarming. That's disturbing. And from a standpoint of we actually see the molecular chaos, like you were saying earlier, associated with this. And a lot of people that are in the field, that are that are in genomics, they say, well, there's no possible way that can happen, because it's not going to reverse transcribe into. No, that's not true. So here there are other pathways besides reverse transcriptase and line 1 retrotransposon, and there is the DNA integrity issue of breakage in DNA. We named in a paper that we put out the the other implications that can actually predispose to integrated events occurring, and reverse transcriptase is not the only route. Okay, repeated ROS related stress on the internal machinery of the cell predisposes the DNA to the possibility of an integrated error to occur, right? So
So
we believe that yes, it's integrating into the DNA.
terrifying thought and likely fact. So that is still a completely separate issue than the spike protein. Right. And so, like, what we're seeing with spike protein is that there's no, for one, there's two parts to this question. One, how are people being exposed and having elevated spike proteins that weren't? Was it simply the virus? Is it shedding? What do you think there? And the duration of the spike that is exposed. I've seen one study that was over 700 days, right? Like at extremely high levels of this consistent spike production. So, what are your
Yeah.
thoughts on that?
Well, when you get the exposure to the virus, naturally you're going to start developing antibodies to the spike, and then retention of that spike. I've actually seen studies that are showing greater than 3 years of retention, and the thing is that if the retention is active, and it's because some viruses go into what we call dormant expression, right? So a dormant expression is that a person then they're not subject to maybe symptoms until their immune system becomes compromised in some way, they in an exposure. So then all of a sudden, boom, it's like this whole cycle begins again. So there is a certain type of dormancy behavior in viruses, and synthetic SARS-CoV-2 does have a dormancy to it as well. Some people who have higher what we call susceptibility based on their HLA type expression. There are some people that are more susceptible to higher symptom based problems. Like, in other words, some people would get more severe, have a more severe case and expression with some of these sequelae that we see, with more complications in the systems, like with the heart, the brain, etc. And then some, their HLA susceptibilities are a little bit better, so where people are not having as much, right? But what we see across the board with the mRNA vaccine is, across the board, the people that have been vaccinated and boosted, they're not doing well at all. They are actually having some very significant expressive changes. And a lot of people, they don't realize that there may be associated changes with that until they start having some real health effects that they have to pay attention to. But the thing is that the spike retention will continue to be the case because the spike is retained in the tissues, right? And then people that are getting vaccinated, we're only going to boost that effect. So any boosting that they get is going to boost the pathology. It's not going to boost resilience, it's going to boost the pathology,
Wow. Okay.
right?
Okay. So you've connected a few things for me, and it makes sense why now that the cancers they're different. They're completely different now, and they're not responding in the same ways. They're coming
Right.
back with vengeance. They're coming out of nowhere. The relapses, right? And some things have clicked. So one thing that I focused on pretty early on was the SP40 promoter or enhancer, right? But you laid out a whole nother aspect of it. But the big question here is going to be, to my knowledge, we've not seen the evidence that SV40 is actually in the Moderna like it is in Pfizer. So, but we're still seeing people have problems with both, right? Like, and so that's one difference. So, what are your thoughts on that? Why is Moderna still doing the problems that we see with just mRNA without the SV40 issue?
Yeah, this is a really great question from the standpoint of shared motifs, and the engineering between two different companies, and what's disclosed and what's not disclosed. We're also seeing it from the standpoint of high possibility of cross contamination in the manufacturing process, to where you have vaccines that are manufactured in the same plant, so they might be in a different section, but they're sharing the same groundwork of the manufacturing process. So there's high risk for cross contamination there. Then you actually have people that were said to be receiving a Moderna vaccine that received a Pfizer because someone reached in the refrigerator and grabbed out the wrong, the wrong lot, or they grabbed out the wrong vial. I mean, there was no look early on in this whole process. The controls were terrible, so cross contamination risk is really high all the way around for those parameters that I just mentioned. You know,
Makes sense. It's yeah.
yeah,
Um,
yeah.
okay, so on if you get on anything where you're looking into long COVID or in any sort of turbo cancers, you're going to come up with some sort of a spike protein detox, right? And so with that, there's one out there that's been extremely popular, I would say, but what Dr. Catanzaro would like I'm not seeing it work. And so that is concern. So it's a combination, curcumin and enzymes that are supposed to break down the spike protein, but not seeing it work. So what are your thoughts on that, and how are we going to have hope to at least address that side of it? The whole DNA side seems pretty scary. We got a lot of work to do there. But on the spike protein side, how do you feel we can best address or mitigate that?
Yeah, I think that, of course, a really thorough molecular understanding and surveillance of the person's, molecular landscape to find out what is being, affected and dysregulated. That's the first thing. These anti-spike protocols, they're used, in general to assist with controlling inflammation, controlling, immune responses, and helping with bolstering up the body's health process. And and of course I've used a lot of these things for years in integrative practice, and they do work, but they are not, in the spike sense, they're not clearing the spike, they're not mitigating efficiently against the spike, and you can't expect them to do that because we're talking about a very accelerated issue. We're talking about a translation issue that's happening in very fast. How can a single agent possibly address a very high gradient of discrepancy, a discrepant signal in the system? These things are helpful, but they're not completely mitigating the process. So now, if a person was taking them, I would say that it's important for them to bolster up their immune system and take the things that they know are going to assist their immune system to get stronger, like things like nattokinase, lumbricokinase, quercetin, curcumin, vitamin D, a lot of these different things that you know about, or even the physical modalities, even like, for instance, people are doing EBU to try to clear their system of, what we call remnant inflammatory elements. I mean, these things are helpful, but whether or not they're clearing the spike completely out of the tissues and expression in the interior of the cell, unlikely, unlikely, right? So there has to be a mitigation strategy that is going to directly affect controlling and mitigating against the high rate gradient of discrepancy that's happening in the cell. So you have to have a precision targeting effect that's not going to aggravate the mRNA initiated aberrance activity, right? So the faulty activity that's occurring.
Th this whole situation is absolutely psychotic. Like, just think about this. We injected people,
Yes,
billions
agreed.
and billions of people with mRNA. It's not even a vaccine, right? To create a spike protein that now we have an absolute catastrophe on our hands, and we don't know the best way to go about it. Like, that's psychotic.
It is. It is, and that's the reason why people have been, talking about this is indeed a bioweapon, because of the fact that this is exactly what we're seeing. It's a very chaotic process. It's a very uncontrolled. There's no off switch to it. And believe me, look, mRNA is a natural messenger RNA is a natural part of our body. Synthetic mRNA is not. Recombinant mRNA is not part of our body. So our body has natural mechanisms by which messenger RNA works with a natural rhythmic movement to allow for what we call a protein-to-protein cross-talkability and stability. So in other words, it maintains the integrity of our functional proteins. So the functional relationship of our proteins depends upon a stable transcription translation. That's part of what mRNA, natural mRNA's responsibility is in the body. Synthetic mRNA is from the outside that corrupts that message, right?
So let's just go out on a limb and say Albert St. Georgi was correct in that proteins have an electromagnetic structure. And then we take something and we just alter all the proteins within our body, right? That goes back to that whole chaos aspect, does it not? That we're
Yes, it
altering
does. Uh-huh. Yeah,
everything.
absolutely. Everything. So in other words, it's not just one segmental issue. So in other words, it's not going to just say, oh, well, it's just going to be hitting this one area. No, because it's creating what we call a broad, a broad disturbance, where you hit one protein. You got to remember, we talk about protein to protein interface and crosstalk. Crosstalk is a big word, by the way. Crosstalk means that the communication properties of protein to protein interactions to get collectively to maintain, functional control. So when you start dismantling that process, your functional control is it affects every system, not just one area, right? It affects every system. So
I think
that's the
you've
reason why
laid
you
out
said, like you say, it's
everybody.
chaotic. It is chaotic. It is a very chaotic thing.
Well, I think you've articulated why it's been so frustrating for essentially all practitioners, right? And everybody's on different levels for like not knowing what to do with this specific situation because it is a massive, massive problem, and it is so many people, right? It's so many people that are seeking help. Going, we're trying to turn over every stone and understand it. So thank you for laying it out. I hope anybody that like hears this understands how serious it is. But I want to throw in just a little bit on one thing I feel like we can do that absolutely every single human being has the possibility is photobiomodulation, right? Like those type of therapies, which my favorite would be the sun, right? Get in the sun. And going back to what you laid out with the electroterrestrial chain mitochondria, like the only way to really repair those systems in the grand scheme of things is infrared and UV light, right? That's how we repair these heme proteins that have been absolutely annihilated with these mRNA shots. It's just unbelievable. So, like, how do you feel about that? Is that something that at least we can try and hope that it's going to
Yeah,
make things
most
better?
definitely. Any of the natural source, ability for sunlight and getting your body into a good fitness state, where the adaptation mechanisms are bolstered. These are all, from a biophysical standpoint, are very important. So any modality that encourages a better, electromagnetic interface for better cellular strength, because as you know, every membrane of our cell has a polarity principle, and those polarity principles need to be enhanced, right? If you're going to be able to endure and have less breakdown, a bottom line breakdown in the systems, your cellular controls have to be really strong and not vulnerable to high stress, likelihood for risk for mutations to occur that are negative mutations, right? So these things that you are mentioning from a mechanical standpoint and a physiological standpoint are essential. I mean, these are essential things to have as a base. And like, let me just preface this too. Not everybody is ready to hear this message. People that have been vaccinated are terrified that they made a wrong decision. And then we're not coming on here to say, oh, you should have not done that, and I blah blah blah. Many people were pressured into it. Many people did not know about what was happening, and the transparency was not there. So there's a lot of people that are suffering, and they did elect to have this vaccine because either their jobs required them to have it, they would be losing their careers if they didn't do it. And look, I don't want to go off on a rabbit trail, but not everybody's ready to hear this message, but please keep an open mind because of the fact that this thing that they're promoting, by the way, is still being promoted in 65-year-old and greater people being vaccinated and boosted up to 8 times is a dangerous, very dangerous thing. So please keep an open mind about this because of the fact that there's, to add more boosting into your environment is to add more insult to pathology, and it's going to be a terrible thing, right? So, but again, not everybody's ready to hear this message that I made a major mistake in taking this vaccine, right?
Well,
I
you're
mean,
right, and
it's
you're spot on, but, like, at the same time, like, this conversation is so unbelievably important for likely the practitioners, right? And even those
yes,
that haven't gone down this rabbit trail, and this is the reason why it's so frustrating, why we're not getting the results, and why things are we keep hearing the same talking points over and over, and things are getting worse and not better, right? Even for people that haven't taken more boosters, they're not getting better. I'm not seeing it at least. And so this is
no,
why, I'm
no, definitely not. Not according to our side either, Logan. We're seeing more and more cases coming in that have very high, disturbing, complicated disease processes that are initiated by this. That's what we're seeing on our side.
So the
And
goal is to try
you
to
know,
get, solutions, right? Like, because nobody, you can't be browbeat on this, and you can't go back and change something that we've already done or not. So what I want to tie this, and I think it's probably going to sound a little bit like a tangent, but I'm going to bring it back. So the latest thing with acetaminophen and autism that came out. This is the new thing. Like, depending on when you're listening to it, that was just announced by the Trump administration, Kennedy, that uh, increases the risk of autism. What I'm was interested in, because you have the immediate argument, well, I took Tylenol and my child didn't have autism, right? So this is where I'm going back to that. So I went back and I started looking and trying to figure out the probabilities, because I this this is a little bit tangent. I don't really believe in cause and effect. I believe in probabilities, because the same person can do the same thing and it manifests in a different way, right? And this goes into redox, heteroplasmy, and all that. So when I was looking at something that would have an embryological effect, I came across, and I was familiar with, thalidomide, right? And so, like, not every woman that took this anti-nausea medicine had a birth defect in their child. It's actually only like 20%. And so what we're finding is that week, it's like uh, it, roughly 20 to 30 weeks, right in there. This is like the big important time. If you took it there, it was a higher risk of having these birth defects. So what what I'm saying is that the timing, that lot number, the biophysics, when you look at thalidomide specifically, part of that problem was based on chirality, right? Left and right-handed molecules altering optics and everything else. So when we're talking about spike protein, we're talking about these injections. Is that timing and chirality of what potentially a spike protein could be causing be part of this as well?
yes, absolutely. So the chirality and the actual structural relationships of what we're seeing in the actual engineering itself is a very vital, part of the action that will elicit the mechanisms of action, albeit the undesirable actions, right? So, so we remember, because we got to go back to the whether it's S1 or S2, foreign matter, foreign synthetic matter coming from the outside. And as you know, the body always has to make a continual adjustment to anything that it's exposed to environmentally. Well, this is an environmental toxicant. Okay. Let's say it for what it is. It was developed in the lab. It's a foreign material. It now becomes what has been termed a bioweapon. It is a environmental toxicant that is actually created significant disruption in the intact mechanisms of the human systems. And so So therefore, we are dealing with an entity that has a very high unpredictability to it. So, and we've been seeing that system wide. Why some people actually have more cardiovascular microclotting, why some people actually have more neurovascular over cardiovascular, why some people actually have more kidney dysfunction involvement, why some people actually have some significant biome disturbances. It doesn't manifest the same way in every person because it's a very unpredictable toxicant.
Yeah, it's absolutely terrible. So this is
Yeah,
where you just talked about this foreign body, right? Is what makes me so unbelievably concerned about supplements, right? Because when you understand the big one was like, and I don't even know how I came across looking at and it may have actually been on raw milk when I was fighting over, the world over trying to get raw milk in Arkansas and looking at Louis Pasteur is the one that actually found chirality when he was looking at grapes, wine.
right.
And so w then that led me down this trail of if we have any sort of a synthetic, right? And then so starting to try to tie in like MTHFR and folic acid versus methylfolate, right? Like what are the optics doing in these different mechanisms? I think all of that is really valid questions that I don't know that anybody has the answer for because I'm not sure that we've really looked at it, right? So because we know on that top level we can have certain emission absorption spectrums and they differ, right? Methylfolate and then folic acid actually have different absorption and emission spectrum. So when when we start looking at this, I'm very, 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 very concerned by most of the protocols I see because what that is it's take a vitamin C, take zinc, take vitamin D, right? Like this is kind of that immune boosting thing, and I think that is so misguided and so absolutely detrimental when you factor in what you and I just talked about.
I would agree with that. And I would say that when you're looking at a more precision approach, you know, understand the underlying root cause. I mean, all go back to that root cause. And the bottom line is first do no harm. Understand what the root cause is. And when you're understanding the root cause and what the expression is, then you can intelligently take the steps to be able to bolster the areas, not from a shotgun therapeutic approach. Like, in other words, you can't just throw everything at something. You got to be a lot more strategic than that and triaging your ability to get the systems that are affected and really enhance those systems from a standpoint of resilience. You don't do it by shotgun controls because shotgun therapy or shotgun nutritional replacement, or shotgun pharmacy. You actually have to be able to know more precisely what is going on. And it's very important to do that. And from a biophysical standpoint, that's where biophysics comes in. The biophysics and the mechanical aspects of our engineering and how our natural environment interfaces with our ability to stay healthy or what we need to do to adapt better, right? It all actually comes to the core expression. So that core expression of adaptation. So if you're adapting in a very positive fashion to where your body is accommodating function versus functional loss, right? And the more we can enhance the function and from a natural rhythmic point of view and from a precise view, the better off we're going to be in being spot on without trying to throw everything at it. Because there are some things, even if they're natural, you're going to be putting it in there, but maybe that there may be totally uh, not really effective because of the fact that you're not understanding the landscape very well, right? So
It, it, which the supplement thing is a moneymaker and it's very easy to promote and see ads and be inundated with things, right? So what you have said has actually solidified my beliefs more so, is that the uh, circadian cycles and sun are probably one of the most important things absolutely everybody can do, right? Like infrared and complex 4 creating metabolic water that's going to help line that out, right? If we have excessive reactive oxygen species, like how do we mitigate that? Well, we probably need to maximize our natural melatonin production, right? Like we need to see the sunrise. We need to have dark nights. We need to not be destroying melatonin with all the blue lights, all the non-native EMFs. What you're laying out concerns me on a whole nother level because we do know, I mean, this is Nora Vocal's work, that non-native EMFs, cause tears in the blood-brain barrier, right? They reduce that. So like now we're letting all this crap into our brains as well, and that's a whole nother level. So like for one, I just said all that to say, like, I think if getting in the sun isn't part of somebody's protocol and getting good quality sleep and embracing circadian just efficiency, then I don't think we're going to move the needle. I just think that that's the foundation it has to be. But I don't know if you agree with that or not. But the next level of this is we got a lot of sick people that are going to need results. So with that, you're with Neo7, that's your baby, that's your business. And y'all have done incredible work. You've published in papers. You're continuing to be detective and practitioners. So what are y'all currently working on? How can we at least have hope that at least we have stuff coming down the pipe? Because a lot of this is just going to be absolutely terrifying for people. So what is that hope that you're working on? And am I right on at least saying that,
Yeah,
circadian
a lot
is
of hope, in getting a lot more, in tune with what really is happening, from the standpoint of how a person is affected by these negative hits. So in other words, we're not just like out there saying, okay, we got a diagnostic tool. We're going to tell you what your problem is, and we don't know how to fix it. No, what we're doing is we're learning from what the expressions are to create a more precision approach to allow a person to recover, to have the chance to recover by using natural type of progressive, like in our sense, we're using polypeptide engineering that's directly matched to the patient's expression or the person's expression to assist them in being able to have better regulation. But that's not the only thing. I mean, that actually creates the molecular surveillance creates the ground substance by which you can look at these systems and say, oh, I can see that I need some attention in these areas. And you can actually take your modalities, whether it be physiomechanical modalities or, simple things of getting out, like you're saying, uh, when the sun comes up and getting that fresh air in there, getting that exposure, changing your routine around for better R&R, better sleep, all of these things, nurturing yourself, all of that proper nutritional and lifestyle type clean way of eating, which you've talked about many times. I mean, these are things that are so important to be able to get a person to recover. The hope is to become more precise at the ability to understand what's going on, finding the root causes, and addressing them more intelligently, and not just taking pop shots at them, and not trying to layer in longevity biohacking onto somebody that is struggling, because a lot of these people are then guilted into, well, you need to be biohacked, it's because your body is aging rapidly. No, it's because you've been exposed to something that's bad. And yes, I do agree that you need to have the longevity factor addressed, but the primary focus at hand is to get the functional ability again, to allow your body to recover and to build toward resilience. You can't just biohack resilience into somebody that's layered with multiple complex signal interferences. I mean, it doesn't happen. They're already compromised. So you're not going to layer in longevity on a compromised human being. You got to help them regenerate and recover, right? I mean, it's common sense here is, okay, doesn't mean that resilience and longevity protocols wouldn't help them. Oh, yes, of course they can help, But the point is that you can't biohack something in that's going to just automatically shift the aging process in favor of reducing your body age, right? It doesn't work that way. People think it does, but it doesn't.
It's kind of like being worried about getting your car detailed when you got a busted axle. It's like that. That's completely missing the problem of why you can't drive. So, what what do you think has the highest level of hope specifically that somebody can just take away from after they listen to this conversation?
Yeah, the hope is probably number one is when you hear this information, don't run to terror and fear as your first reflex, because the whole purpose of disclosing the problem is to allow for a better intelligent understanding of how to approach to the solution. So it's very important, because when you run to fear and terror, you start grabbing at everything, things from a panic point of view that are main that look, they've already unleashed the panic on everybody by putting the mRNA vaccine out there. So you certainly don't want to be responding in that posture. So the first thing is get grounded and realize that whatever is being revealed is to take a more intelligent, precise approach in getting to the solution. And then when you get there, you realize that it's never about the single thing you do. It's about the combination of things that you need to put into practice into your life. Whether it's get, like I said, all the natural things, and better sleep, sun exposure, good ways of eating, clean ways of eating, rest and relaxation, whatever your personal enrichments are, spending time with people who make you laugh, and these type of things. All of these relational things are important in being able to create that robust health picture, right? A lot of the times when you do this, you're already past the 60% mark in recovery, because you're putting all of these great ingredients in your body process, and you're already there and posturing into a resilience. The rest of it is coming up with a precision hub strategy and building your triage around you to continue to help you to move in that direction from the standpoint of what's going on internally, addressing it, and moving forward. And some of those elements that we talked about earlier, the integrative elements like some of the nutrients and some of the vitamins and some of these other things, they all can come into play as long as you understand where your system deficiencies are. Find out where your system deficiencies are and start bolstering them up in resilience. Get a hub strategy. And that's what Neo7 Bioscience is about. Not that I want to have a commercial here, but Neo7 Bioscience is about finding the errors that are happening and creating a mitigation strategy for resilience going forward as a hub, right? And this we've been seeing across the board has been helping a lot of people, because they're not navigating blindly. They're actually understanding what is affected, and what they can do about it. So there is hope, right? There is that hope.
I'm definitely rooting for y'all. Praying that y'all hit on some things that are needle movers, and we're gonna
And
keep
Logan, we're doing ongoing research to not just uncover the problem, but also to find the better engineering forward. So in other words, we're not just trying to look for the problem. We're trying to help people to understand the nature of what this is. And it's a very hard thing, because some people don't like hearing it. And when that comes into play, you've got a larger entity that's financing the big machine of mRNA technology. And when you actually have that big machine being financed with multi hundreds of billions of dollars, then there's always the problem of being able to you're coming up against that. And of course, their message out there a lot of the times is not a good one, basically.
Apologize for the abrupt ending to that one. We ran out of internet or something, just went completely out. So we covered a lot there. I hope that it was not just scary, but extremely, extremely important as we continue to look for solutions. There's a lot of people working on this, so don't be too scared. Just know that like we need to focus on what we can control. Like with Lander, I said, what can I control as daddy? What can I do for my baby? And focus on those things. And I really hope that whether you follow me on Facebook and or in our group, it really is the basis of natural circadian aspects. So getting in the sun, the morning sun, being out in the sun in the middle of the day, getting UV light, where we're producing vitamin D, where we are doing the things that build natural production of these hormones and molecules that are really crucial to overcoming this absolute catastrophe that we are currently in. And I think that we're gonna keep figuring out things. It's gonna come together. I don't know exactly how, but I think that there's a lot of things you can do, and we've got that. So thank you for following along with what we do. I hope it adds value. The podcast. If you're looking for a place to come and have these discussions, Dr. Petra Davlar and I have the Sowing Prosperity support group where we meet every week. You're welcome to jump on that. Just sowingprosperity.com, and you can find out all that information. And we got a really good group and have these discussions. And so have a wonderful, wonderful rest of your day.